The background behind today's primer session is that I had a lot of people who were asking me how to do differential gene expression analysis in bulk RNA sequencing data. And so that pushed me to port a script that I had developed locally and set it up for sort of more widespread distribution on Terra. So I've been working on this, script for a decent number of months at this point, and I've been using some version of it for the last couple of years. It's a pretty robust setup, and I just want to say I'm not gonna make this workspace private or anything. This is a public workspace, so everyone should feel free to copy this script over to their own Terra workspace. And then if you're having trouble getting things to work on Terra, because it's not perfect, and I'll show you where I tend to have issues, just in case you want to use Terra for other things. This should work locally if you have RNA-seq data locally, or if you're attached to one of the Samba shares, such as helium or iodine. Helium is the one that we use for MedPop AFib, or MedPop rather. Okay, so what I've got up, because the screen share is up, is the cloud environment configuration. This matches the PDF. So I do want to hit the edit button pretty soon. But yeah, this is the cloud environment configuration. So 16 CPUs, 60 gigs of memory. And then for your application configuration, you want it to be set for R bioconductor. So of note, what we're working in is called an IPython notebook, or if you've been around for long enough, or yeah, it's called an IPython notebook if you've been around for long enough, and a Jupyter notebook if you're more recent to this type of Python interaction. And so they now support R as a language. So this is a Jupyter notebook, and which is typically used for Python, or has long been used for Python, and we are using this with the R language set. So there's a couple of different options in Terra that I won't get into, but we're specifically using R for this. Okay, so I'm going to close out this cloud environment window, but this image is repeated literally twice. And so you just want to get this cloud environment started. It may take you a couple of minutes. Hopefully, most people have gotten that going. And again, if you're not able to exactly perfectly follow along with what I'm doing, this is pretty well documented. You should be able to accomplish it locally, if not on Terra. But please do ask questions, and and I will stop and try to address everything as best as I can. But I also tried to design this code to be very much something that you can do without somebody walking you through it. So we'll see how today goes. It's going to be an interesting test. Okay, so hopefully you're in the process of getting your cloud environment running. Mine is running. I started this a couple minutes ago. It'll take about 2 minutes to get up and running, and you'll see a little sort of double arrowed circle here. And I'm seeing a lot of questions in the chat and the Q&A, and so hopefully I hope that means that people are getting going. I'm gonna hit this edit button.
There's one question, if Terra has an RStudio version.
No, but they're talking about it. And that was right before COVID hit, that I was talking to some of the folks that are involved in implementing Terra widgets. So that conversation didn't get that far, but there's not an RStudio implementation. So yeah, for the record, I write everything in RStudio, and because it's the only implementation of R I feel really comfortable with, and then I ported this. So yeah, I didn't just write this blind, doing that in just the R console is a little bit weird. But I think that this is much more like an R markdown document, what we're working with today. So good question. And I'll reach out to the Terra folks after today's lecture and see if we can, see if we can get some clarity on that. But I know that the COVID stuff has really thrown everyone for a loop. Okay, so my notebook is copied over and into the cloud environment, and we're ready to start. I'm just going to give you guys a little preamble. What we're going to work with today is mouse left ventricle data from mice that have had a gene called CANT2 knocked out. It's ubiquitin SCF. I guess it's like an E3. I'm trying to remember this exactly, but it's like an E3 ligase adapter protein, and it's one of the loci that's involved in atrial fibrillation. So what I've done beforehand, and the reason that this is all commented out, is because this is what I did on Prem. So I use Callisto on Prem to do the initial analysis of this data. So this isn't like a fully Terra functional. I do quantification of the RNA-seq data, which we talked about last week. on-prem, and then I use the Google Cloud SDK, the utility gsutil, to copy that to the Google bucket that's associated with this notebook. And you should all be able to use the commands that I've set up to copy from this Google bucket into your own specific Terra workspaces. So hopefully everyone has gotten or is in progress for copying over this workspace and has gotten this workspace going. And again, if you fall a little bit behind or you wind up getting to this after the fact, don't worry about it. You can shoot me an email if you wind up with any issues, but it should be pretty self-explanatory. So what we've copied over are 3 wild type left ventricle bulk RNA-seq data sets and 3 knockout left ventricle bulk RNA-seq data sets. And we'll do a little bit of comparison of how those data sets look. Hopefully we'll get to that part. And then we have a file, this tximport sample LV. It just has 2 columns, sample and condition, and the samples are just these names, and the condition is just wild type or knockout. So pretty simple experiment design here. And then I also have, I used to be able to use the utility Biomart with Terra. Our image underwent an update over the last couple of months, and now I'm having a little bit of trouble with getting that to work properly. So I'll show you where that is. It's very easy to accomplish locally, but I basically made 2 files, one that goes from an ensemble versioned transcript to the external gene name, and another that goes from the external gene name to a gene ontology term. And so those I've uploaded so that we don't have to worry about, basically pinging the server and winding up with errors. But those you should be able to accomplish locally in our studio pretty easily. Okay, so this is a clean environment for me. I actually did clear everything out so that I'll be working from the same state that you guys are. But the way that you run code in Terra is once you've clicked that edit button and it's copied it over to your cloud environment. So this is environmental setup. So we'll just hit run. And you can see that the star here indicates that it's running and then it's completed. So this is the cloud environment setup we have here is pretty robust. You should find that things complete fairly quickly. This will take us a few minutes, which will hopefully give us some time to catch up or catch any issues. I'm going to start executing. So we're installing these libraries using base R. I was having some trouble with the install.library, but install if missing as a convenience function works really nicely. And so you can do that for everything that's not a bioconductor package. If you're not familiar with bioconductor packages, or if you're not familiar with bioconductor, it's a really great resource. It's a really great community of people that work predominantly on analysis of biological data. And this is everything from microarrays to methylation sequencing, all kinds of stuff in R. And so we're initially, this bioconductor is installed on this version. You can actually see that here. They say, BioC version 3.11.1. So we're actually using bioconductor here. We know that it's already installed, which is really nice. And so we're basically saying, okay, bring in the bioconductor manager, and then we're using the manager to install these 8 packages.
Amelia, there was a question as to whether you generally install every time you run or if you use a Docker image.
So what I typically do is I actually cleared out my cloud environment last night so that I would actually be installing with you guys, and that would give me a better sense of pacing. But generally, I don't save off Docker images for this. That's a good question, though. I've been thinking that I kind of want to get into that, and I just don't feel familiar enough with saving off my own custom images that I've done it yet. But I'm thinking that might be the next step for me. But once you have them saved to a workspace, it keeps it in that cloud environment. So I don't typically clear out my cloud environment, more than once every so many months. You only really have to do it if you're, if you want to do something different in that workspace or if the base image updates. But yeah, great question. I think that's something I'd like to get more familiar with and just haven't had the time to. Okay, so this is still running, and you can see that it'll take us a couple more minutes, but you can see it's actually, we're already down to the A general linear model package, which is our third to last. And it's just installing dependencies for everything. So this is I think it's cool. This is really pretty fast. for, you know, the fact that you're paying like less than $1 an hour. So we're going to get a lot done for, you know, everyone's 77 cents.
There was a follow-up question for that as to whether it means you keep the virtual machine running or the hard drive running. So how is it that you continue your session?
You can shut down the cloud environment and it'll maintain the steady state. And I think that what people are referring to is that there's, you know, a persistent disk cost of a couple dollars a month. And so that is, so the paused cloud compute cost is like almost nothing. It's very low in terms of the monthly cost. And then the disk cost is like a couple dollars a month. So this is basically what I'm paying for when I shut down the cloud environment, and then I keep, you know, data in a workspace. Because for the last, you know, 3 weeks or so that I was, you know, retesting and redeveloping and adding the gene ontology content to the script, I did typically keep the cloud space open, and I would only really zero it out when I was like, okay, this is a workflow I want to test from start to finish and without any, you know, priors in place. So yeah, great question. And it's, you know, I think that if you're on this and you don't have, you know, a billing account through your, you know, through the Broad, and you're just on the free account, you know, it's not bad. I think they give you like $250 in free credits, and that's enough to do quite a bit of analysis. So given the persistent disk cost is, I mean, there's not a lot on this persistent disk either. What will transfer over from the Google bucket is like, you know, I don't know, a couple 100 megabytes. It's Callisto data is pretty, it's pretty small once you've actually installed it. The initial FASTQ data is quite large though. So yeah, this is probably the slowest step of all of this. The rest of this goes pretty fast. Okay, so hopefully, and it sounds like, okay, it sounds like I'm hoping that everyone is, you know, getting through these initial steps okay. Always a little bit like, oh, is it going to take 5 minutes or 6 minutes? But we're probably all hitting Terra at the same time. I did warn them. But yeah, the first, I guess 2/3 of this are probably the most stable because I've been using these for a while. And then what I've implemented here is TopGo, and then TopGo as a dependency R graph visualization, which is, it's a little bit wonky. Okay, all right, so this just finished. Hopefully it's finishing or close to finishing up for everybody else. I'm gonna keep going on the next few steps, kind of explaining what we're doing as we go. We've got our R environment set up with all of our packages installed. And, you know, as people have said, these will be persistently on your cloud environment unless you, you know, elect to redo your cloud environment, and, you know, can ideally be, if you use these packages all the time, bundled into a Docker image that can be custom loaded. Okay, so our next step here will be, we want to bring in our libraries for handling. And so I kind of, you know, separate them out. These libraries, I'm going to start running this, but these libraries up at the top, DEseq, tximport, RHDF5, Reader, and Biomart. These are our libraries that kind of like do, and Biomart in this case, I don't even really need to be bringing in, but if you were doing this locally, or if I could get the web access aspects of Terra to work, we would be using Biomart. Then we have a bunch of plotting specific and data manipulation specific libraries. And then a few things that come from TopGo. And then there's a nice little function in gene list that we'll hopefully get to at the end. Okay, so those go in. Okay, so we're gonna make a directory for this quantified data for tximport, and then we're gonna list the contents of the directory in which we sit. You can see that I ran this last night. Okay, so mine says quant tximport already exists, but for you guys, hopefully it won't be saying that because you haven't done this yet. And so the directory that we're sitting in is this bulk RNA-seq pathway. This IPython notebook is sitting here, and then we have a directory quant tximport. So now we're going to use gsutil and manage the directory structure. So everyone get this command started a minute or two, just because it needs to go and copy the data. And then for those who are less familiar with R, like myself, I use it a lot, but I'm much more comfortable with using Python to go and interact with the system but there's a command in our system, and you can literally just give it a command that you would put on the Unix command line. Okay, and so that's actually just finished up. And so it's basically saying, go recursively copy everything from this quant tximport directory and drop it into the local directory quant tximport. And so now we're going to examine the downloaded data here. And so you can see that there's a deprecated file that I use to test things. But then there's this gene to go term, text to gene. That will make this directory called output for everyone in a minute. And then there's several folders that hold Callisto quantified data. And then there's this left ventricle tximport sample. And then so we're listing the contents of the directory quant tximport, and then a space, and then we're just listing the contents of the directory where we are, just to double check. Now we're going to check our working directory. And we're living in this, folder called edit. And then we're going to make this folder called output. And we're going to set our base directory to be in the quant tximport, because that's where we want, that's where the data we that's where the data we want to manipulate lives. So I'm getting this already exists, but you guys should see that it creates this directory. And then we've set our working directory. We're going to double check our working directory, and we should be located in the quant tximport folder. So, okay. So now we get to the meat of what we're actually going to do. So we're going to read in the data that we've quantified using Callisto, and it needs this table. So first we're basically making a list of like a directory tree of where it should go and look for files. And then we're giving each of those, file paths a name, a sample name, which is right here, names for the files right here. So this is a named list. And you need to, this has to return true. And it should for you guys, because this is data that I've quantified and checked. But if it doesn't return true, sometimes the H5 files, which are generated by Callisto, will have an error or like a weird end of line thing. And so you can use the RHDF5 package to actually check and make sure that that is a valid file, which I'm not going over here, but it's something to think about, particularly with Callisto data. Okay, so in this code block, we're going to do an initial intake of our data, basically telling it, okay, we're using the function tximport for files, which is this structure we've made up here. We're saying that they're Callisto quantified data, and we're saying, the transcripts should be included as well. So the thing with this data is, it's ensemble, I used a mouse ensemble genome to index genome to quantify this data, and a mouse ensemble GTF to quantify this data. So this is the part where we'd normally be using Biomart, and I've commented it out, but this will all work really nicely locally. And so what we're doing, just so I can show you guys what we're doing, is we're setting up a mart saying use the mouse musculus, the M musculus gene ensemble data set, use the ensemble mart, keep it verbose, and that's what lets you see these attempting web service request things, which I really like in RStudio, because if you're going to have an error, it will show you what that error is and often give you a clue as to why. And then we're going to establish, so we're actually going to query against all these gene IDs, which is the row names for the object that we just made in the variable counts. And so we're going to basically grab this so that we have a list of all the ensemble transcript IDs for mouse. And here we're contacting the Biomart database, and we're returning a list of ensemble transcript IDs, and they're corresponding, this says HGNC symbol, but it's actually external gene name, because I originally wrote this for human data. So we make an object, tx2 gene, we use the get Biomart function, and our filters are ensemble transcript ID, and our attributes are ensemble transcript ID, and then we'll get also the external gene name. And then the values that we're looking at are the gene IDs, which we just set up right here, and then the mart is the mart that we just set up right here. And then once this returns, and this can take like 15 minutes sometimes, so that's probably part of why it's throwing an error. But then we'll change the column names. The first column will be called TX name, and the second column will be called gene ID, which is actually not that important. I just kind of like things to be organized. So this is how this would work, and this will work locally just fine. But because I wasn't able to get Biomark to work with Terra this time around, we're just going to read in a table that is basically the output of me doing this locally. And that should take just a second. It's a pretty long file. It's like 100-something thousand lines. And now we're going to do the same thing, but the difference here, and I'm just going to start that running, but we're going to do gene-based quantification. So the whole point here is that we're going to go from, many transcripts of the same gene to rolling that up for one gene. So if you want to do isoform determination or isoform-based stuff, you do it a little bit differently. But we're basically just saying, okay, the transcripted gene file is the object transcripted gene. And we're saying, okay, don't give us the transcript out, roll that up by gene. Okay, so now we've got everything that we need to actually import things in DEseq2, which is going to handle all of our normalization and all that great stuff we talked about last week. So I'm going to start this and I'll walk you through it. So and this, again, these all go pretty fast, but here's our object, and we're saying, okay, DEseq dataset from text import. We're handing it the object that we just made it. We're handing it the file that delineates the samples, and then we're saying, look at this based on condition, which is a variable from the samples bulk object. And then we're saying, okay, the condition is a factor, and then we're establishing the levels of wild type and knockout. And so, sometimes it's not going to be wild type and knockout. Sometimes it's going to be, liver and brain, or doxycycline and sham, or what have you. But this is just delineating the two conditions that you've got. Okay, so we've talked a little bit about, QC and data. So I established a keeps file, which is, vector, which is basically saying, okay, anywhere that the counts across a row are less than 10, and these are raw counts, but anywhere the counts across a row are less than 10, we're going to, or greater than or equal to 10, we're going to keep those, and then we're going to get rid of the rows that have a total count of a given transcript or a given gene across a row of at least 10, reads, basically. So this is filtering noise, which can be problematic if you want to study a gene that isn't highly expressed. And I think there's other ways to do that. This is really intended to identify, big signals of differential gene expression. And then we actually use the DEseq wrapper, and then we also set up the results. So we use this function results, and we put that into a file, and then I immediately write that out into the folder output that we made, and then give it, a nice useful name. Okay, so this has gone through. Okay, and so now we're going to identify genes with a log2 fold change of greater than or equal to 2, so we can plot them. So I'm going to start running this, and we'll walk through it. So I'm using the function LFC shrink, and I'm using and we installed the HGLM, the general linear model, in order to handle this. There's other ways you can do it too. If you have a really, really large set of samples, like I process 150 LA versus LV human sample set, I use the transform function rlog instead. But for less than 30 samples, LFC shrink works just fine. So it's just worth keeping in mind, if you're doing something that has many more samples than what I'm doing, you may want to do things a little bit differently. And I'm also happy if you reach out to me to help out on that. Okay, and so we're basically saying, this is base R right here. We're just saying subset this where the log2 fold change is greater than 2. And then I'm grabbing the row names to get the gene names, writing it out as a table. And I'll often just take these names and throw them into a web utility for pathway analysis. You can do a lot of things like that. It doesn't have to all be in R. And then I'm doing the same thing for where we have a log2 fold change of less than -2 and writing that out. Okay, so we've done that. Another thing that's really useful is once we've run the samples into DEseq, we've also had we also have a point where we identify, or where we normalize the matrix. And so playing with that normalized matrix, you can do a lot, without having, R invoked, or you can do a lot of things in other, plotting contexts. And so what we're doing is I'm actually using rlog to do the transformation here, but I could have used LFC shrink. But I'm going to start executing this. And what we're doing is
There is one question.
Yes.
Oh, sorry to interrupt. When you're pre-filtering for to require greater than 10 count step, wouldn't you lose the most highly expressed genes, which weight maybe 0 in wild type? I know this comes up all the time, and I'm always asked about it also.
So sorry, can you, let me,
Oh, yep.
I'm having
So if
trouble
you
parsing. Can you
require
repeat
at
that?
least 10 counts, say there's a gene that's not expressed at all in wild type, but then becomes highly expressed in your experimental condition, wouldn't you lose those genes by requiring at least 10 counts?
Well, it's the sum of the row. So let's say that, you know, your wild type, you have, you know, yeah, let's say for your wild type, you have, you know, 10, 10, and 10, just that, which is a fairly, for decently sequence depth data, that's actually not particularly high levels of expression. And you have 0 or 0.01 or what have you, for your knockout, just to throw an example out, you'd retain that row because your row sum would be like 30. Yeah, it would be like 30. So it's across, yeah, so it's the sum across the entire row. And you're right, this is only 6 samples, so we could drop this to 5, but that's a threshold worth playing with. But it's not, this isn't like an average value. This is literally just the sum across the entire row of the matrix. So you wouldn't lose any gene that's highly expressed. Like if you have a single sample that has a value of, you know, that has a count of 10, you would retain that row. So we don't filter a huge amount. I want to say we filter less than 10% of the total number of genes. But yeah, that's an important point though. This is like a raw row sum. It's not like an average value or anything like that. So we're really just trying to remove genes that are generally not expressed across any condition. So yeah, that's a great question. And I hope that the answer that I gave is sufficient. Okay, so let's see. Okay, so we've transformed and wrote the gene sample matrix to a file. We did it with a log2 fold transformation and without a log2 fold transformation, because you don't always need to be, you know, log2 fold change. Sometimes you want your raw normalized numbers. Okay, so next we're gonna get into plotting, which is pretty cool. And I'm not gonna show you guys the plot yet because it's really cool. I found this package and I'll start running it. Enhanced Volcano, which is really nice. It does automatic labeling, and you do need to be a little bit finicky. I'll show you what it looks like. You do need to be a little bit finicky when you actually output this file if you want it to retain transparency information for editing it in like Adobe Illustrator. But I think that the data that it outputs is really nice. So in this case, what you can see is, you know, what do we have? Like this is the wild type on this side, and this is the knockout on this side. And so we're saying for left ventricle, what are our big changes? And as you would expect, CAND2 is the gene that we're knocking out. So obviously we have a ton more CAND2 expression. We see it, you know, we see it drop very precipitously. We see a massive reduction in the log2 fold change for CAND2. Though actually for the left ventricle, the majority of these changes occur in the atria, which is part of why we're just using the left ventricle as a test case. Anyway, I really like this package, and the person that wrote it is really responsive to questions and wrote really nice documentation. So this is one nice way to display your data. Okay, so this is additional visualization. This is what I like to do for heat maps using the normalized count matrix on genes with a log2 fold change of greater than or less than 2. So I'll start it running. It'll take a moment or two to run. This is all stuff I kind of, you know, spent some time writing myself. I, you know, spent quite a bit of time with the hclust function, which is how we make a dendrogram, which will be the first image that shows up here, where we're plotting hclust for the distance. You have to transform the matrix that you work with, or it'll look really weird. And then to actually plot things for the heat map, I just appended the, you know, the greater 2 names and the less than -2 names that we isolated earlier. And then we subset our differential expression gene list, that we output here. We subset that just using the row names that are in this, you know, LV log2 fold change differential expression list. And so there's 2 heat map generation methods here. This one uses the base R function Heatmap. Heatmap.2 is really nice, and what I like that it does is it produces a density. It produces a density plot, so which I think is a really nice legend. So here's our cluster dendrogram. So as you can expect, we do actually have an outgroup, which we for each set, which we maybe we want to go back and re-examine that. But it's nice to look at the cluster dendrogram to be like, okay, how nicely do these cluster? And there is some distance for a couple of the samples. But we do see that, the knockout samples cluster together and the wild type samples cluster together, which is the big thing that we want to see. And then when we look at the initial heat map plotting, so we don't retain this dendrogram here, whereas with heat map 2 we can add it in. But here's all of our lists of genes that are downregulated that are basically upregulated in knockout, the genes that are upregulated in wild type in comparison. So there's not a lot of genes here, which makes the Go term stuff kind of interesting. But yeah, so I think it's a really nice representation of what's actually changing in your heat map. This was a particularly isolated list. It's almost always quite a bit more genes, and you typically can't even read the names. But this example is really nice because of that. And then I really like the density plot here because I struggle with having a good legend. And I really like the legends that Heatmap, like this, legend density plot that Heatmap 2 produces, especially for preparing figures for publication. So this is my method. And I'll often turn off the row dendrogram, but I think it's interesting. And you can see there's sort of, this module here and this module or this module here. And then it is just a slightly fancier version of what I had shown previously.
Amelia, there was one question just about the difference between normalized counts and TPM or RPKM.
Sure. I think, so for me, the way that I've always thought about it, and I don't know if this is entirely correct, but I've always thought about normalized counts as necessarily being TPM, RPM, FPKM. Because at that point, you have to be looking at transcripts per, in this case, this is Callisto, so this is transcripts per million. And so I think of them as as soon as you invoke weighting functions to basically say, okay, there's a difference in, total sequencing depth between, sample 1 and sample 2, and we need to align them so we can compare them. I think about that as transcripts per million at that point, because you have to be thinking about things in that way in order to compare one sample versus another sample.
Yep.
And then you're using a weight. Sorry?
Oh, and then there's the RPKM, which actually takes into account the length of the transcript, which obviously is not in all normalization approaches.
Yeah, and so if I remember right, in DEseq2 does take this into account. I was going to say we correct for library size, and then up further here, we use, there's average TX length from the assay function, which corrects for library size. And then this does do, one of the reasons that I like DEseq2 is it does take that into account with the dispersion, with the gene-wise dispersion estimates, the mean dispersion relationship, and the final dispersion estimates. And I don't have the details about that specifically in my head, but Michael Love, who wrote DEseq2, has written quite a lot about how to think about that. And so I know that I think Salmon also does that as well. But that was one of the reasons I went with DEseq2 as opposed to EdgeR, because I thought that DEseq2 implemented that in a way that at least I found easier to understand. But I don't have in my head right now like a way more detailed description, other than you do want to think about, for very long genes and very short genes, you want to have a good model to compare them.
There was another question that branched off your dendrogram check that you always do, which is such an important step. And it's relating the experience of having a comparison that was one female mouse that was, and then 4 male mice. Sorry, I'm reading it incorrectly. So 1 female mouse and 4 male mice in the experimental group and 5 male mice in the control group. And then identifying X-linked genes as the most significantly differentially expressed. And so do you recommend doing a leave one out analysis to ensure that there's no single outlier sample in your batches?
I think that's, yeah. I mean, it depends. If everything looks really clean from looking at your initial dendrogram. So like, for instance, I happen to know that all of the mice in this study are male, so I'm not too worried about there being, sex-based discrimination. But yeah, I think that's a really good thing to do. I think leave one out, and then before you even get there, if the cluster dendrogram looks funky, like I've already done that for this project, but just running a PCA plot, which I think is like, I think the DEseq2 function call for that is like literally plot PCA, and then you hand it your object. So I would say both of those are good ways to just assess, okay, are these really the samples that I want? Are these really the most comparable, most consistent samples in each condition that I want to include? And ideally, you want to have at least two, otherwise you're not really getting any variability, in a 1 versus 1 comparison is a little bit dicey. But yeah, I would say either leave one out, PCA, check the dendrogram, and some combination of those should lead you to, if you have any massive outliers that are by sample, and this also applies to, I think, any batching condition, right? If you ran something on one sequencing platform versus another, or did one library prep method versus another library prep method, if you've got that documented, any of those methods will help you isolate the sources of your variation in experiments. That's a great question. Okay, so we've got 10 minutes left, and I do want to see what we can get through in terms of this pathway analysis. Hopefully you guys can hear me now that I'm closer to the router. So, okay, so this is the newest part of this section, and the one that I'm, this is the stuff that I'm least familiar with. But I wanted to have some implementation of pathway analysis because we did go over that last week, but fairly briefly. So my favorite pathway analysis databases are the gene ontology databases, just because I find them to be, sometimes quite obscure, but I find that using sources like Amigo to, examine genes in a pathway and things like that, I find that to be very useful. So I'm going to start executing this, and then I'll walk you guys through what I'm doing. So first, we're just printing the results table, which is really short. Which has a log2 fold change of, greater than 2. So we're just looking at that for right now. But again, it's a short list. So this is mostly a toy example. I think this would work quite a bit better if we had if we were looking at samples for this project where there was like a lot of gene expression change. So here I'm basically making, so I'm making named vectors, which I'm not like the best at doing. So this is sort of where I ran into the most trouble in testing this. But I'm, making using the p-value, the p-adjusted p-value, and I wind up using, basically what I'm doing is I'm setting a bunch of things up to feed to this function that TopGo uses. It's a wrapper function that we'll see in the next code block. So we have to define the gene universe, which is basically all of the genes that we didn't filter out earlier on when we filtered based on row sums. And then we read in this table, which is basically, gene to go terms, which again, I got using Biomart. This is how you would do it. It's commented out, but it's not difficult. You basically just say, give me the external gene name and the go ID. And you'll just get back a list of go IDs and gene names. And then we're converting from table format to list format, just using this nice little piece of R right here. So we're just, converting that over. So here you can see this is what the results table looks like if you haven't looked at one before. And so you can get, numeric p-values and then adjusted p-values. This is I think it's a Benjamin-Hochberg adjustment, so it's pretty robust. And this is just for the log2 fold change for genes that went up. So, and then I just always like to, because I wanted to make sure that they're named numerics. I just check the structure of the gene universe and the structure of this adjusted p data frame right here, which is what we set up in the previous slide. So here's our wrapper function. So we're saying LV bulk go data, and I'm going to start running this. And we're saying it's a new object of class top go data. I'm giving it a quick description, log2 fold change of greater than 2. I'm saying use the biological process ontology. I'm saying all of the genes involved are in the gene universe named vector. And I'm using this function top diff genes, which we actually brought in way in the beginning of this session. and I'm saying, filter anything that has a node size of less than 10. So has like less than 10 connections. So we're filtering out some of the more, I guess, some of the more really specific, but also really obscure Go terms. And then we're using, this is again, this gene to go. I'm basically saying, okay, your annotation functionality is going from gene to a gene ontology term. And here I'm handing you the object gene ID to go right here. And so there's different ways to use this. You can use this with old array data too, which I think is actually what they wrote it for. But I'm not using microarrays anymore, and so we're running this now. Okay, so it finds your specific gene IDs, and you hand it this with p-values included, so it can kind of make a decision about where to do a lot of the filtering, which I think is interesting. Okay, so it finds these Go terms. Based on your gene versus Go term comparison, and then it annotates the nodes. And so what we're going to do is we're going to plot that. And then we're going to do a Fisher statistic test to basically take a look at what came back as significantly enriched. Which is, it's like there's not that many genes involved here with a log two-fold change of greater than 2, so it'll be interesting to see. I'm also using some options to control my plot width and height so you can see this better. The text is still not great though. So don't be too concerned when it's a little hard to see. All right, so I'm going to start running this.
Amelia, there was a question for TopGo.
Sure.
You can either use KS or hypergeometric for enrichment analysis. And do you have a preference for which one of those you use?
I think KS is good. So you can see that these are grayed out here. I ran into some errors when I was running that and didn't have enough time to. I really wanted us to do a comparison between KS Classic and then the elimination method and then Fisher. I think Fisher is like nice and simple, but what I like about the KS test is it's similar to GSEA. And so I think it's more robust. We're doing this with Fisher because that's what I can currently get to work on Terra. But I would say, the KS test is a bit more robust. But I also don't want to give a specific recommendation without, knowing exactly what your conditions are. Other than I think that this is, really well validated. And Fisher is a little bit more, basic. Not necessarily bad, just basic. Okay, so we've gone through this. We've generated a table of the results, and it's always interesting what works and what doesn't and how long it takes to optimize things. I gave myself like 5 days to get this package up and running. And it was, enough to get a basic up and running, but not everything, unfortunately. And the variant here, I'm using their function show sig of nodes and just saying use the score result Fisher, use the 5 for significant nodes and then use all of the possible info. So it's, there's quite a lot of nodes here. That are non-trivial. And so it's basically saying, okay, here's what I've done, here's what I've output. There's 764 terms that have a p-value of less than 0.01. They don't recommend using adjusted p-values, and they give a complex rationale for it. Most of the pathway analysis tools I use do have adjusted p-values, like Panther and Enricher. So that's one, or Q-values. So that's one where I still don't feel confident enough in exactly what I'm saying there to say you should definitely go with this versus not. It's more just this is a first pass. And so it's hard to read these terms, but they get more specific and more significant as the colors get darker towards the bottom. And I think, yeah. And so if we actually output what all results looks like in this little data frame, we'll see that we're getting some pretty specific terms back, like circulatory system development, which is good because this is cardiovascular data, structure morphogenesis, these are young mice, and then, interesting for me, actin filament-based process, muscle system-based process, these are terms that are sounding more and more like something involved in, cardiovascular development. Okay, so lastly, but not leastly, this is just a whole bunch of playing around with ggplot, with the ggplot geom call setup, just to do some basic bar plots of the Go PP enriched terms. And you can do these with a custom palette, which I couldn't. I had it working, and then I changed the data, or changed the structure of what I was doing a little bit, and it unfortunately stopped working in terms of it's supposed to color the p-values by how significant they are. And I didn't have quite enough time to troubleshoot it, but it's not too difficult to do. So this is just going to have the rainbow style plot. So I'm basically establishing, just to walk you guys through that before we have to end, making it nice and big so we'll actually see a decent sized image. And I'm saying, okay, you know, I'm doing a negative log transform of the classic Fisher so I can get a log minus 10p in order to actually plot these. And then I'm setting that as a factor, and I'm setting the levels to the, you know, to the levels of the log transform p-value. And then I'm setting the term as a factor, and setting the terms in order of the p-value, and then I tried to do one where I set it as numeric, and that didn't quite work. So it's not perfect, but it works reasonably well. With a little bit of work, it'll be very pretty. And then this is just me using ggplot, basically saying, okay, plot the term on the x-axis, plot the log transformed p-value on the y-axis, use the Fisher value as a fill, though that doesn't actually matter because we got rid of the scale fill manual. But then, you know, setting up some labels and labeling our axes. So this is the, you know, the log minus 10p right here for classic Fisher. You can see the p-values are actually labeled here with the most significant through the least significant, but these are all quite significant. And yeah, that's pretty much the last thing I've got. And, you know, then we can, you know, output all the session information. So we use the development tools to output the session info. And that's really nice because that'll let us know all the information about this session that we ran, you know, exactly what's going on. Like this is R4.0.2. I normally use 3.6. So if things are behaving a little bit differently, that might be why. But that's pretty much what I've got. And seeing as it's 9:20, that's about the time we have for today. So yeah, shoot me an email, ahall@broad. If you have any questions, I'll try my best to answer. And this is just, you know, a quick view through what I typically do for RNA sequencing, you know, differential gene expression analysis and some of the plotting that I do to support that. And I hope that it's been helpful. And I'm, yeah, happy to take any final questions if we have time. And I'm going to leave this open so anyone who wants to access it should be able to access it, you know, basically forever. We've got it set to public, and we're totally okay with sharing this data as a test set.
Thank you so much, Amelia. That was an amazing presentation, and it's a great reference to get to have this code for people to use. There were just two quick questions that I'll ask before we take our break before the MPG session. And one was just getting back to that KS test within TopGo. Do you feed in the log fold change or the Q value when you do that test?
Yeah, so we basically, let's see, we feed it, oh, you're saying for the test? Okay, so so basically any of these tests, right, you know, the run test functionality, that's all based on these Go objects. And so, yes, when we feed it the gene universe, and yeah, this is more or less what we want to be focusing on. When we feed it the gene universe, we are feeding it, you know, the value, or yeah, we're feeding it the adjusted p-value from the results table from DEseq and the name of the gene in question. And then when we feed it the data frame, yeah, so we're feeding it a similar thing when we feed it or actually, yeah, we're not giving it this directly. We're giving it the gene universe, and we're saying find the top differentially expressed genes. So we're giving it specifically I'm giving it specifically the Benjamin E. Hochberg adjusted p-value when it works through this object, and basically letting it do its own selection. But you can delineate your own selection mechanisms as well. But what they recommended you hand it was the gene name and you know, some p-value. I chose you know, somewhat aggressive adjusted p-value for all of for your total results table from DEseq2. But obviously you could filter this.